Okay, fine. So first I would like to express my gratitude for this organization of the first virtual meeting of the Academy, which is being a big success. So, and I would like to start by expressing my sorry for the bad news of this morning that Mario Molina has passed away. As we all know that we're gonna miss a lot the presence of Mario Molina because, but I'm sure that his example will always be present in your academy because he was one of those scientists that embraces problems that had a global importance. And I'm sure that he was very worried about all this situation and he had a lot to add to this. Okay, so I don't want to frustrate anybody, but I'm going to talk a little bit about science or what we're doing to improve possibilities to treat people. And we all know, we hear this 1,000 times, that this pandemic is been showing a big social injustice for many reasons. And we know that after all, we're going to be left with a big problem. The things goes away, we're going to have a vaccine, but we're going to have a lot of leftover to take care. And therapeutics in this sense, especially the ones dealing with infection or the respiratory tract, may be important, and that all the alternatives has to be well looked at and see how can we come up with solutions for the world in general, because we all know that even we having problem already to deal with how we're going to distribute, how we're going to afford vaccination of all the world. And this is because each of us live in a different economic reality, and solutions for the problem has to take that into account. Okay, I am in Brazil. I am in University of São Paulo. We have a center that uses optics for everything. We use optics to study atoms. We use optical to produce atomic clocks. We use optics to do health science. And is called Biophotonics. And this is what I'm going to talk today. What kind of perspectives can we have? Do we have evidences that we can do good treatments? Sorry, with biophotonics. Biophotonics is basically, this slide kind of explain everything. It's the use of light either to control metabolic reactions to improve the cell efficiency or to kill. And in case of cancer, we've been killing a lot. And we worked for many, many years, more than 2 decades now, in developing techniques that can treat cancer. But we can think about infection as well. If we can go near microorganisms and produce reaction induced by light, we can decrease infection in situ or maybe systemic. So my group goes from basic principles to experimental models and to clinics. And some examples I'm going to give to you today. We already reaching clinics and related to respiratory infection, microorganisms resistant to antibiotics, and this kind of thing. Okay, so there is some issues that I should place. Is the cost of healthcare. You know, I live in a country that the cost of healthcare is a problem. You know, even though we have a social medicine, the government is supposed to provide care to everybody, but this doesn't happen the way that we would like. And the reason that doesn't happen is because the cost of health is really blooming, it's going up. And people are very worried about this, because we know that science never had so many good contributions for healthcare. But on the other side, those techniques, those new advances has been never so little available for population in general when we talk about the whole world. So we have to worry about this when we come with solutions, because if we come with solutions that will be prohibitive for most of the population of the world, we are not really solving the problem of the crisis. Okay, mainly what I'll be talking about is the fact that after the infection, there are many, many consequences. We hear every day different consequences on the neural system, on the vasculatory system. We had a very nice talk yesterday. On the liver, on the kidney, but we know that respiratory part is always going to be a problem there, and we have to deal with this. I just put some numbers here. We'll be speaking about number. I compare Brazil and the world, and I see that we are near about 10% of the world cases, which is a very big worry for us in Brazil. But I see that the death rate, you know, when I compare the number of people that are contaminated with the number of people that die, is about the same everywhere. So those are the numbers presently. I took it this morning from the sites, and it's very scary, right? We are already on the 35 million people, and we are already 1 million deaths. And people claim that until the end, we're gonna probably double this. So this is a situation that we should worry about. And on the top of this, the other infections did not stop. We still have the resistant bacteria, and they are growing. Believe it or not, we are very worried about the COVID, but resistant bacteria to antibiotics are growing. And we saw yesterday a few, in fact, I even asked about companies and their position. And we saw some slides showing that by ADA, show a slide showing that companies are not launching new antibiotics because they don't have option. Well, so this is a problem because very, very, very much like many people that contract COVID, not all of course, but a good fraction of them will be debilitated for a period at least. And then they will be target for pneumonia. And if they get resistant pneumonia, we are facing the same problem. We don't know what to do. And we gotta come with different solutions. I show you that we have today 1 million people dead because of COVID. But we have 700,000 people dead every year because of the resistant bacteria. So it's not very far apart. So this is also a pandemic, the resistant bacteria. And so far, very little solutions is being proposed for that. Okay. Well, my colleagues, academicians, know that I am a fanatic for this photodynamic action, which is a technique that I use to apply to many things, and we've been improving a lot. And the idea is a molecule that can be synthesized or is a natural one. Sorry. So if we can put this molecule near the microorganism or a cancer cell, I deal a lot with cancer. When we excite by light, this molecule is nothing, it's not aggressive for anything. But when it's excited by light, it produces a reaction with oxygen, which is present in almost everywhere, especially on us. We are not, we are aerobic creatures or animals. So anyway, when that is transferred to oxygen, you produce a very oxidative type of oxygen. There are two types of oxygen. The singlet, which is the dangerous one because it oxidizes any density of electron you have. And as you know, we are made of biomolecules with a lot of electrons making the structure. And the triplet, which is the good one that participates on the respiratory cycle and production of energy in your cells. So the idea here is then putting molecules near the infection or distributed, make sure the microorganism acquire this and illuminate, try to put the infection down. This is the principle that works, and this is the same principle works for cancer. I'm not going to speak about this, but being a success in the world, and even in Brazil, the social medicine is now considering adopt those techniques for social medicine. Okay, all these infections that you can see in this plot is being treated with the principle that I just present to you. And here in Brazil, we've been focusing a few, especially respiratory tract, And the condyloma, which is also a problem, is the second cause of death for women in the world, is the papillomavirus that evolved to cancer, to cervical cancer, and many others that we've been dealing here in Brazil. From my group, we already helped to launch a few products that today is available for society. But I want to concentrate in the infections that is related to the present situation. Well, I hope that Francis is in the audience because 2 years ago, we proposed some techniques that's being demonstrated to clean up organs for transplantation. Normally, the donor may be with some diseases like hepatitis or maybe coronavirus. And if we can clean up organs for transplantation using those optical techniques, is a good gain. And we've been demonstrating this, and this in Toronto is being applied already for the transplant of lungs for donor patients with hepatitis. Okay, so this shows you the powerful that this kind of mechanism it is. And this is already published in the Nature and in the Lancet 2 years ago. But now we are focusing in the infections that are consequences or are the most common ones that contribute to the increase of the resistant bacteria's resistance to antibiotics. Throat infection, that the name is of pharyngotonsillitis. This pharyngotonsillitis is common everywhere, and we all have to deal with this infection using antibiotics. You could think about a kind of pill that you put in your mouth, takes 5 to 10 minutes to dissolve. This molecule goes to the plaques that is infecting your throat. The microorganism acquire these molecules, and then we go and we eliminate. And then we could decrease a lot the infection without the abusive use of antibiotics, because many throat infections, they are not responding so well. It's not uncommon people that have almost permanent throat infection. Okay, so we have been collecting the microorganism of common pharyngotonsillitis of people in general, and we demonstrate in the lab that this technique is very efficient to kill all the microorganisms that normally cause throat infection. And then we did the preclinical studies, and now we're doing the clinical studies. And the whole idea, let me jump a few slides, in the way we develop new devices for the doctor to really map the infection of the throat using optical techniques like fluorescence. He actually can have a better view of the conditions of the infection if he uses fluorescence image. But this is not the case. What we are developing now, and we are doing a big clinical trial now, is produce this molecule in one of those forms, either a syrup or a tiny caramel, but it has no sugar on it, or a pop. I think the name in English is lollipop. And then the kid, we want to focus in kid, but we got the permission of the government for going to adults in the big clinical trial first. So this is a way to put the drug on the infection on the throat. So that is about 5 minutes of using this. I've done that, immediately comes the illumination. And the results is fantastic. In 94% of this is a pilot study with 60 young kids with the pediatrics, and this is adults. In all of them, we solve the problem of 94% of the, or 90%, roughly speaking, of the infections. We all know that antibiotics takes about 48 hours to start to act. This acts immediately. And from 10 to 24 hours, the infection consequences like fever and other symptoms goes away. So we are now in a big clinical trial with adults. And we've been discovering many things, including that about 11% of the bacteria on the common people throat are resistant. So we are already facing a big problem in this sense. And the idea is we are now doing this clinical trial, so we have to collect material for everybody. And the result is that actually the people that do, or I'm gonna show only one of the symptoms, which is fever. For the people receiving this treatment, in less than 24 hours, the symptoms of fever and everything goes away. Now, if you take antibiotics, this is displaced. Of course, many antibiotics works well, but it's displaced. And we know that antibiotics, the abuse is causing problems. So this is a way of that we are now implementing here. But there is another one that's new, and this is very nice. Imagine that you have pneumonia. It is possible to make those molecules to go down to the lungs and to go to the colonies of bacteria in a way that with the extra body illumination from outside, we can reach the colonies and kill the pneumonia. And the answer is yes. At least it's very efficient for the small animals, which is what we did. We use the pneumonia in the little mouse, and then we made them to breed these special molecules. Here we are using endocyanine green, and now we are using nanotechnology, and we make this to go to the lungs, to impregnate the colonies of bacteria, in this case, in pneumonia, right? And it's amazing that all the rats that we treat, they survive. The ones that we didn't. This is the Kaplan-Meier survival plot showing the efficiency in rats, or in mouse, I'm sorry. But now we are preparing to go for people, and we are starting to do the security. And we already show in animals, small animals, security. We do not do any damage to the organ. We only damage the colonies of bacteria in the lungs, which is a good thing. And now we are even improving the way to delivery the molecules to the lungs by breathing using nanotechnology in a way that will be very efficient. Well, we are preparing to do people together with Canada and here in Brazil. But before doing people, which is the same mechanism, you know, you have pneumonia, you breathe, and you do the illumination on the thorax chamber, and then we should, in most cases, get free of the disease. Equipment is being built, but we are doing now pigs because we have to understand a few things before. And many people say, well, we are not transparent. I know we are not transparent to visible light, but to infrared, we are quite transparent. And when I illuminate the thorax box, many light go through with the infrared. And this is the light we're using. So those are experiments with pigs, where we show by seeding microorganisms inside the thorax box chamber, that we are able to kill efficient all of this. So you see, in a few minutes of illumination, we can reduce many log scale of the microorganism we have. So I believe this can be a very good technique for treating pneumonia in general. And now we are investing a lot in introducing technology like the nano-emulsion and everything together with collaborators all around the world to make efficient breathing of molecules so we can treat well. Because the secret here is making the molecule to go deep in the lungs. Well, we're doing many studies, I'm gonna jump, but then there is a problem that the Canadian colleagues put to us, which is the treatment of cystic fibrosis, which you know is a problem, and there is very little solution, even though medicine is improving a lot, the quality of life of this. This is an infection on the lungs, it's genetic, you know, you get from your parents, but we believe that we can deal with this infection. Well, I'm not going to speak about cystic fibrosis, but what I want to tell you, I'm going to jump this. What I want to tell you that medicine did a good work in improving the average age that a guy with an individual with cystic fibrosis can live. But as you can see, we are living out. You know, we are leveling out. We are condemning these people to live no more than 36 years. And it's a problem because then after that, infection became even, well, there are more complications in this disease, but we want to deal with the infection. And I think this will be a good target and everything. There is another problem in the present situation, which is the use of ventilators. And we all know that ventilators, when you get artificial respiratory action, you get infection, because that strange object on your trachea is a nucleation of colonies of bacteria. So most part of the people that gets to be intubated, they have to get antibiotics right away. But the problem is they are in hospital where the amount of resistant bacteria is huge. Can we help with that? Yes. Imagine you can have a tube which is functionalized with a special molecule here from curcumin, in a way that we put together with the tube a tiny little fiber. As you know, an optical fiber is like a hair. I can't give an example, but many people can hair. This is the dimension of an optical fiber. So if we put this together, we don't mix it up anything with the respiratory action of this tube, but we prevent the infection, because this light will inhibit the formation of colonies of bacteria. So in blue here is the evolution of the amount of infection in one of those tubes if you do nothing. And underneath in red is what's happened when you're doing this procedure, which means that we inhibit the evolution of the infection, which can be a good thing, because most part of the people you intubate and people die of the infection they acquired.
Vanderlei, please conclude.
Well, okay, I will conclude, but I was speaking about resistant bacteria. You know, I told you they are huge, and companies already said that very little solution with antibiotics. And then we'll be doing a demonstration that we can do the photodynamic to really kill these bacteria. And we select the bacteria. And the question is, will photodynamic revert to the situation of the resistance? And the answer is yes. If I give a shock of their new generations somehow loses a little bit the necessity of high doses of antibiotics to be killed. And then we did an experiment where we measured this minimal concentration for inhibition of the bacteria, using, of course, bacteria that are resistant. And we demonstrated that by using some concentration and some shock of photodynamic, we can make bacteria that was previously resistant to some antibiotics in some level now became non-resistant. Okay, I guess the president is very excited that I finish. So we are also cleaning up the plasma for bloody, because as we hear yesterday, one of the therapeutics for COVID is taking the plasma for a patient that had. It has the antibodies that will help, but everybody wants to take out any virus that are there. So by separating cells and plasma, we can somehow deal with this problem as well. So, well, this is my conclusion. If before we could be concerned with fighting infections, now this is essential, right? Because we're gonna, the post-COVID, the post-crisis, we're gonna have to deal very much with this. And I think photodynamic therapy is a, photodynamic inactivation is a very good possibility. And the cost benefit of this technique is tremendous advantages, right? This treatment costs no more than a good dinner. So it is affordable for many, many parts of the world in terms of technology, in terms of cost. Thank you very much. And we'll be trying to produce the molecules in large quantity, because this is what's necessary for this. Well, I would like to thank your attention and all my collaborators. There are too many, and but at least some of them are here, and I thank you all.